Hello children, welcome to biology class. Today we are starting a new lesson, cell cycle and cell division. Last day we complete our discussion about the lesson cell, the fundamental unit of life. Today let's discuss about cell division and the cell cycle. Children, we know that all the living organism in the entire world starts their life from a single cell. Yes, what is the name of that single cell, children? Yes, zygote. So this is a zygote. Okay, so growth, and you may wonder how a single cell then goes on to form such large organisms, isn't it? Zygote is converted to embryo, then to fetus, then a new organism is formed. And not only that, growth and reproduction are characteristics of cell, indeed of all living organism. All cells reproduce by dividing into two, with each parental cell give rise to two daughter cells. And what happened to these newly formed cells? They again divided and a new organism is formed. And also in growth, when those organisms, their growth is completed, repair is going on. So for that, our cell is following cell cycle. Okay, here in this picture it is very clear. G1 phase, S phase, G2 phase and M phase. Okay, so let's discuss in detail about cell cycle today. So this is a cell cycle, children. Cell division is a very important process in all living organism. During the division of a cell, DNA replication and growth. It is very clear in this picture. DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way. For what? To ensure correct division and formation of progeny cells. Progeny cells means daughter cells contain intact genome, isn't it? Being Homo sapien, we know we are having 46 number of chromosomes. So in newly formed cells also they should possess 46 number of chromosomes, isn't it? So it should ensure during cell division. So the sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell and eventually divides into two daughter cells is termed as cell cycle. Children, you have to study by heart this sentence. Okay. Then although cell growth is a continuous process, DNA synthesis occurs only during one specific stage. Okay. Then the replicated chromosomes are then distributed to daughter cells by another stage. These events are under genetic control. So now let's discuss the phases. Now let's discuss the phases of cell cycle. A typical eukaryotic cell is illustrated by human cell in culture. So these cells divide once in approximately every 24 hours. The diagram is there in our textbook. However, this duration of cell cycle can vary from organism to organism and also from cell types to cell types. For example, can progress through the cell cycle in only about 90 minutes. So first of all, let's divide the cell cycle into two phases, G1, S, G2 and M phase you can see in the diagram. So we can divide it as interphase and M phase. These are interphase and M phase. Okay. This one is M phase. M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phase. So that is this. Okay, and it is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour, isn't it? The interphase lasts more than 95% of duration of cell cycle. Then the M phase starts with the nuclear division, and it is explained as karyokinesis, and the division of cytoplasm is cytokinesis. Let's discuss the difference between karyokinesis and cytokinesis. Now, cytokinesis is the division of the cytoplasm of cells. Cytoplasmic division, cytokinesis. What is karyokinesis? It is a division of nucleus. Clear? Then cytokinesis occurs at the end of M phase after the nuclear division is over, whereas karyokinesis occurs during M phase of the cell cycle before the cytokinesis begins. Clear, children? Now let's discuss three phases which are explained as is very clear here: G1 phase, S phase and G2 phase. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Okay, that is G1 phase. And during this G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. Clear? That is G1 phase. Next is S phase or synthesis phase. It marks the period during which DNA synthesis or replication takes place. Children, replication means unwinding of DNA. We know that DNA is a double-stranded structure. So, for the synthesis, it has to be unwind and new single strands formed. And from that strand, new strands again formed in the daughter cell. That process in detail we will discuss in class 12. Okay. So, keep it in mind. In S phase, synthesis phase, what happened? DNA replication takes place. During this time, the amount of DNA per cell doubles. Okay, and if the initial amount of DNA is denoted as 2C, then it increases 4C. That means it doubled. That means there is no increase in the chromosome number if the cell has diploid or 2n number of chromosome at G1, even after S phase. In G1, if it is at 2n, even after S phase, the number will be 2n. Clear? And in animal cell, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. Whereas in G2 phase, next one, G2 phase, the proteins are synthesized in preparation for mitosis while cell growth continues. Clear? So, and keep it in mind, children, some of the cells in adult animal do not appear to exhibit division. Example, heart cell. And many other cells divide only occasionally. So, as needed to replace a cell that have been lost because of injury or cell death. So, these cells do not divide further, exit G1 phase to enter an inactive stage called Q0 stage. Q0 means quiescent stage. Okay, quiescent stage. Then of the cell and this stage remain metabolically active but no longer proliferate unless called on to do. So, depending on the requirement of the cell. And next one is M phase. We know that this is the most dramatic period. So, you see, here you can see within M phase there is prophase, metaphase, anaphase and telophase. So, it is said to be a dramatic period of the cell cycle involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called equational division. Why it is called equational division? We will discuss it about in next class. Again, but keep it in mind, mitosis is an equational division. Though for convenience, mitosis has been divided into 4 stages as prophase, metaphase, anaphase and telophase. Then it is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Okay. So, the 4 stages, about the 4 stages we will discuss in next class, children. And which are the 4 stages of mitosis? Prophase, metaphase, anaphase and telophase. Okay. So, till that you read thoroughly. Okay. Study well, children.